Today's lecture will be on the organizations of tissues within the central nervous system. In particular, we're going to be looking at the neuroglia or the central nervous system glia. There's structures such as the microglial cells, the astrocytes, the ependymal cells, and the oligodendrocytes that are considered to be the central nervous system glial. These neuroglia provide a couple of important factors. They're either going to protect, they also help nourish neurons, they provide an organized supportive framework for all nervous system, and collectively these glial cells are going to account for half of the volume of the nervous system. Some important key differences between glial cells and neurons are going to be that glial cells are going to be able to divide, or they are capable of mitosis. They do not transmit nerve impulses like neurons, but they do assist neurons in many of their functions. So the first one we're going to look at in the most detail is going to be the astrocyte. The astrocytes are the largest and most numerous neuroglia in the central nervous system. Their main job is to maintain the blood-brain barrier. This means that compounds dissolved in the circulating blood in the capillaries do not have free access to the interstitial fluid of the central nervous system of the brain. First that we see is these paravascular feet here. These paravascular feet are the extensions of the astrocytes, and they're going to wrap around each individual capillary that is coming into the brain. The astrocytes are then going to secrete a chemical, and these chemicals secreted by the astrocytes are maintaining the permeability characteristics of this inner layer, or what we consider to be this very inner layer of a capillary, which is called an endothelial lining. So their job is going to responsible for maintaining the permeability characteristics of this endothelial lining, not allowing certain ions to cross over into interstitial fluid. So this endothelial lining is going to control the chemical exchange between the blood and the interstitial lining. They also form a structural framework. We can see this here, how one astrocyte, the paravascular feet, is connected to a neuron, it's connected to an oligodendrocyte, it's connected to the capillaries. And it's also important that you understand that these astrocytes are packed with microfilaments, and it helps form the cytoskeleton to strengthenize and organize the nervous tissue in the central nervous system. So just providing a basic structural framework. Astrocytes can divide in a process called astrocytosis, which produces new cells to fill in areas formerly occupied by a damaged neuron. So if this neuron would become damaged, the astrocytes would divide through astrocytosis and produce new cells to fill in the area of this damaged neuron. Astrocytes also assist in neuron development. Astrocytes in embryonic brain promote growth and interconnection of developing neurons, so it assists in the development of the neurons to make neuron-to-neuron connection. And as mentioned before, they are going to control the interstitial environment, especially in the brain. They adjust the composition of this interstitial fluid by regulating the concentrations of, say, sodium, potassium, or carbon dioxide in the interstitial fluid. And then they also have the ability, through allowing rapid transport of any type of nutrients, ions, or dissolved gases between this capillary and the interstitial fluid, and including into the neuron itself. So the ependymal cells will go in detail with the production of cerebral spinal fluid. But the first thing that we should know is that these are ciliated cuboidal epithelial cells. These are going to line the ventrals of the brain and also, in this example, the central canal of the spinal cord. They are going to help produce cerebral spinal fluid, and we'll go through that in the lecture on the brain. The cerebral spinal fluid is important because we need cerebral spinal fluid to be circulating continuously. Also, cerebral spinal fluid is going to provide a protective cushion. It helps dissolve gases, nutrients, and waste. So we see these epidermal cells that are located along the central canal of the spinal cord, or we see this in the ventricles of the brain, and they are just going to help or assist in the production of cerebral spinal fluid. They also collectively are going to form, when we have a group of epidermal cells grouped together, we consider that to be called a choroid plexus. So the microglia is going to account for the smallest percentage of glial cells. So these are going to migrate into the central nervous system. So after the migration into the central nervous system, their job is for protection. They're going to engulf any type of cellular debris or waste products or any type of pathogen that's located inside of the interstitial fluid of the central nervous system. And finally, the oligodendrocytes. We can see here that it has a central nucleus, and then these individual processes are in contact with the exposed surfaces of the neurons and involved in the process of myelination. So you can see here with the oligodendrocytes, it is producing myelination, or these myelin sheaths around these individual axons. These myelin sheaths are concentric layers of myelin, which help increase the propagation or increase the speed at which the action potential can travel along the axon.